Okay, so let's jump right into a really fascinating biological puzzle. We usually think that for a cell to grow and thrive, more of the essential stuff is always better. But what if giving a cell too much of a good thing is actually just as bad as not giving it enough?
Right. This is biology 101. It's one of the most basic rules of life. Before a cell can split into two, it has to copy everything, literally its entire contents. It needs to bulk up and gather all the resources for this huge job.
So you would think that giving cells extra building blocks, like guanine, which is crucial for making DNA, would make them divide faster, right? Well, get this. The data shows the complete opposite. As researchers cranked up the amount of guanine, the rate of cell division just plummeted, eventually grinding to a total stop. More was definitely not better.
And that is our central puzzle, a classic case of when more is actually less. So what in the world is going on inside these cells?
It's a pretty wild question when you think about it. Guanine is absolutely essential for life, so how does having too much of it suddenly turn it into a poison that stops one of life's most fundamental processes cold?
To figure this out, we have to look a little deeper. It's not just about the amount of ingredients the cell has, it's about the recipe. And it turns out, the key isn't just having enough of everything, but having it all in the right proportions.
And that brings us to this key idea, nucleotide imbalance. A cell's DNA is built from four main ingredients, A, T, C, and G. For the whole DNA copying process to run smoothly, the cell needs a balanced supply of all four. If you just flood the system with way too much of one, it's like dumping an entire bag of flour into your cake mix. The whole recipe is just ruined.
And here it is, our first huge clue. When the researchers looked inside the cells, they found that adding all that extra guanine didn't just raise the guanine levels. It caused the levels of adenine, another one of the key ingredients, to totally crash.
So why does that happen?
Well, the cell has this really smart system to make sure it doesn't make too much of this stuff. When levels of one nucleotide get high, it sends a signal to shut down the factory. But in this case, the flood of guanine from the outside tricks the system. It slams the brakes on the entire production line, which stops the cell from making both guanine and the now desperately needed adenine.
And this is the proof in the pudding. If the whole problem is an imbalance, way too much G, not enough A, then what happens if you add the A back in? Will you fix the recipe? And just like that, the cells start dividing again, happy as can be. It was the imbalance all along.
Okay, so we found our culprit, this toxic nucleotide imbalance. But where exactly in the cell's life, where's the scene of the crime? Where does the whole system actually break down?
Well, the data shows us exactly where it happens. See all those dots? Each one is a cell. And as you add more guanine, you can see the cells start to pile up in this one specific area. That's the S phase, the part of the cell cycle where DNA gets copied. It's a full-blown cellular traffic jam.
Just imagine a car factory, right? The assembly line is humming along, but suddenly it runs out of left side doors. It's got tons of right side doors, but that doesn't help. The whole line just stops. And that's exactly what's happening in the cell. The DNA copying machinery shows up ready to work, but it can't find the balanced set of parts it needs. So everything just stops.
So we figured out that the cells are stuck. They cannot divide. And this leads to what is maybe the most surprising, most counterintuitive finding of this whole study.
So let's just ask the obvious question. If the assembly line is stopped and no new cars are being made, has the factory itself stopped getting bigger? Common sense would say, yeah, of course. But in biology, common sense can be wrong, and it is, spectacularly. This is what researchers call the great decoupling. While cell division comes to a screeching halt, cell growth, the process of just getting bigger, it just keeps chugging along. How is that even possible? Well, it turns out the cell's main growth supervisor, a really important pathway called mTORC1, is completely blind to this nucleotide problem. It never gets the memo that DNA replication has crashed and burned. So it just keeps shouting, grow, 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 while the division machinery is just sitting there, completely stuck.
And the result is, well, it's pretty wild. The cells, since they can't divide but are constantly being told to grow, become enormous. They can swell up to 3 times their normal size, becoming these huge, bloated cells.
And let me be clear, this isn't just some weird little quirk. This is a breakdown of one of the most basic rules of cell biology, that growth and division are supposed to go hand in hand. It's a critical vulnerability that was basically hiding in plain sight this whole time.
So, what does this all mean? We followed the clues, we've uncovered this incredible twist. Now let's put it all together and really understand what this blind spot is all about.
So let's just recap our little detective story. We started with a mystery. Why did extra guanine kill cell division? The investigation showed us it created a toxic imbalance of ingredients. That imbalance caused a massive traffic jam when the cell tried to copy its DNA. But the big twist was that the cell kept growing, totally unaware of the problem. And that's the final revelation. Cells have this huge blind spot. They literally cannot sense that their nucleotide supply is dangerously out of balance until it's already too late.
And this quote from the researchers themselves just sums it up perfectly. The cell doesn't realize there's a problem until it's already in deep trouble during S phase. And this fundamental flaw just shatters that normal, healthy link between growing and dividing.
And that leaves us with one final and really important question. Now that we know our cells have this hidden weakness, this blind spot, you have to wonder, how might this very flaw be exploited by diseases that are all about unchecked growth?